Welcome to Sketchy. We take all the super complex stuff you need to learn and turn them into memorable visual stories packed full of everything you need to know on test day. Click the link in the corner or description to try for free for 7 days. Now let's get to it. In this sketch, we'll talk about gluconeogenesis. Gluconeogenesis is a metabolic pathway that makes glucose from non-carbohydrate precursors. And what better way to show gluconeogenesis than going to a pirate candy store? And not just any pirate candy store, but one with all those classic characters from Pyruvates of the Caribbean. The purpose of gluconeogenesis is to maintain normal blood glucose levels in a fasting state. Any metabolic disease that affects the major gluconeogenesis enzymes can cause hypoglycemia because we can't make glucose from anything that's not a carbohydrate. Lactate, derived from anaerobic metabolism, alanine, glutamine, derived from amino acid catabolism, and glycerol, derived from triglyceride catabolism, are the main substrates for gluconeogenesis. The spoiled milk, aloe juice, and igloo pie represent lactate, alanine, and glutamine respectively. Lactate and alanine are directly converted to pyruvate. Glutamine enters gluconeogenesis via alpha-ketoglutarate, a TCA cycle intermediate. Just a side note, all TCA cycle intermediates can turn into glucose except for acetyl-CoA. We drew glacier mint gum behind the DHAP treasure chest to show you that glycerol enters gluconeogenesis via DHAP. All amino acids except for leucine and lysine can turn into glucose, as you can see in this diagram. Some of the amino acids are directly converted to pyruvate, while others are converted to any of the TCA cycle intermediates, except for acetyl-CoA, of course. But all roads lead to glucose. Odd-chain fatty acids can also turn into glucose via succinyl-CoA. Even-chain fatty acids can't convert into glucose because they can only produce acetyl-CoA. Please watch our fatty acid oxidation sketch to learn more.